Thank you very much for the introduction and having to speak after two talks speaking about antimicrobial resistance and the burden of them, and both of them in direction of a problem. So everyone's defined the problem. The question is, are there any solution that we can think of that may be slightly out of the box? 2020, I moved from England to the UAE, and I was tasked with at the height of COVID to establish a wastewater surveillance lab. So from a clinical lab to start working with sewage. One doesn't know which of the two is more difficult, but sorry, yeah. So the history of wastewater surveillance is quite interesting, in that historically, if anyone's read Victor Hugo's Le Miserables, he's got a very nice quote where he quotes that a sewer is a cynic. It tells all. And even before Victor Hugo, the Romans, they had the mouth of truth, which is basically a manhole cover with a legend that says if you put your hand in it and you don't say the truth, it will bite your hand off. The classic thought of wastewater has always been that it's a source of environmental pollution. But we all know that it's been providing a lot of information. Anyone who passes through London would remember the story of the cholera outbreak and Broad Street pump with Jon Snow. And during the wastewater pandemic, it was all over the news for COVID-19 surveillance, whereby quite a few governments started using it for surveillance. And it appeared again in the news with the polio outbreaks in London, in New York, and in other places. So it's been used for quite some time, but sporadically and as and when a problem happens. So the question is, why don't we do it continuously in real time, without bias, looking for the known knowns, known unknowns, and unknown unknowns? To utilize the system with the aim of providing early warning, of providing data, and getting stuff by, let's say, bypassing the complexities in having to work with humans. As we all agree that is very complex with regards to governance, with regards to ethics, with regards to consents, with regards to a lot of regulations in and around. If we can identify a method whereby we can collect a lot of information in real time, without being intrusive and without causing people problem, which is cheaper, why don't we do it? So the interest has increased significantly globally to look at wastewater. So historically, I mean, the first one, as we said, was documented was Jon Snow's Vibrio cholera outbreak in Broad Pump, 1940s, and ever since, polio monitoring in wastewater has been utilized and used. And with the advent of molecular methods, more and more sentinel labs are using wastewater surveillance. And I think now in the UK as well, they have it in following the London identification in London sewage systems 2 years ago. It's also become standard methods. Moore, again, from England, in the 1940s, used it to look for enteric bacteria. 1960s, we're using wastewater surveillance for adenoviruses, Norwalk virus surveillance in the 1980s, flu in around 2010, 2011, and of course, COVID in 2020. So it's been a continuous method. We knew about it, but we have never utilized it in public health at a high scale. And I think that is because quite a few challenges exist in implementing these surveillance systems that we have to address. If you think of it, with every single flush of water, the matrix changes, the content changes, the dilution changes. So standardizing the data or standardizing the methods is not an easy thing. In particular, you might have a lot of your biomarkers degraded. You have identifying the infectivity based on shedding, how much does each carrier shed in terms of viral load or bacterial load or infectious load, and extrapolating that to a population. Sewage systems are very complicated, so not all cities have maps that are up to date to show the populations, the water flows, and support the models that could be used to facilitate accurate representation of the data. And there's also there aren't any standardized methods. So if you look at what the EPA, the Environmental Protection Agency in the US does, it's very different to what is done by the Standard Methods Society in England, by the ECDC, and a lot of the work is done in research labs. In England, a lot of it is done in Public Health England, and there's a lab established in the Imperial College in London. But again, standardized methods are not available. So there isn't a single standard method that you can refer back to, which is and has been validated in all the other different labs. Then there's also difficulty to provide data on the pathogen's life cycle. At what stage has it been there? Is it part of the biofilm? Is it endemically available in the sewage? So there are quite a few challenges. However, it still remains a very significant method that now is coming into play. So there's a lot of pathogens of concern that have been monitored through wastewater with their associated diseases. So we've got Campylobacter, Yersinia, Escherichia, and the list goes on, with significant health impacts. What methods that have been classically used in wastewater-based surveillance? We have the standard classic culture methods, which are quite cheap. They're robust. A lot of people can do them in standard labs. But the problem is you cannot detect, again, inactivated or dead organisms. And and it's relatively very slow to and costly in terms of manpower to be used routinely. Of course, everyone's using PCR now for nearly almost everything, but the problem is you are unable to discriminate the viable, again, and non-viable cells. It's used for SARS-CoV-2, for enteroviruses, hepatitis A, and quite a few others. There's the DNA microarray technologies, which makes things a lot cheaper actually when you start using them, but it's still relatively much more expensive. And the method is not widely available in most standard labs, so it will require an investment. And you have the rest of them, so the LAMPs, the FISH, pyrosequencing, and digital PCR. But what I want to stop in is whole genome sequencing with shotgun metagenomics. So I think the method is gaining much wider acceptance. It will facilitate a comprehensive analysis, and is with the advent of supercomputers and pipelines, we are finding that this will probably end up becoming the way forward soon. It has been used to detect and sequence the variants of SARS-CoV-2 directly. There's work again done in the UK and elsewhere with for SARS-CoV-2 showing the ability to differentiate directly from sample to answer very quickly the different variants much quicker and before samples are detected in the clinical populations. And like everything else, there's a lot of, as mentioned previously by Professor Anger, is the chip microfluidics and chip-based screening. There is ongoing validation. We're doing some of it ourselves here in our lab that we established in the UAE, a high throughput lab to do all of this. So we're currently working on validating microfluidic-based methods with a multiplex-like methodology. So one sample, multiple targets in one assay to get an answer very quickly. But again, going back to the initial problem of the sample standardization with a very difficult matrix, locally we've identified our own sort of method of pre-concentration, which others have also validated elsewhere in other labs in the US. But it really is a very, very tedious process, whereby you can either pre-concentrate by centrifugation or by inoculation, milk flocculation, filtration. For us to make this a much more acceptable method of surveillance, we believe and are working towards getting a sample to answer, just like we did with your PCRs for COVID, what I like to call the Nespresso method. From a sample directly into the Nespresso pod and an answer. The uptake of this will be slightly slower. We did a lot of work also while we were looking at the for the whole genome sequencing, to utilize it for taxonomic profiling. And one of the big, the major cost implications is how deep do you need to sequence your sample, and whether you go with 5 gigabytes or 10 gigabases or less than that to generate meaningful information using shotgun metagenomics. And one thing that we've identified is actually it doesn't make that much of a difference with regards to the sequencing depth. The loss is quite minimal. And why would that be important is because of again, it brings us back to speed and cost if we decide to move to taxonomic profiling using whole genome sequencing. And so a lot of the work required a look back to what's done and how it's done and where it's done. And again, every single time we start looking, everyone agrees that it's a good method, but no one tells you how to do it. So there aren't any standardized protocols or methods, not for bacteriology, not for drugs, not for chemicals, not for toxins, for wastewater, that are agreed globally. The next thing is the introduction of the new different technologies of utilizing what's available and feasible in different environments. We did a lot of work looking at the 1950s method, which was developed in the 1950s by Moore for identification of salmonellas. And we sort of agree with a lot of other scientists working on the field that it's very cheap. It just involves a cotton wool swab that you keep hanging on the water for 24 hours, and then you do direct targeting by PCR molecular, and we end up with very similar results, like when we get composite samples using autosamplers. We looked at the storage conditions, which will differ from country to country, place to place, in terms of degradation, in terms of sample processing and time to reaching the lab, which again requires further looking into. We looked at automation, and we established a fully automated system here in our lab, but not many people can afford to do that in less privileged areas, in societies or governments that are probably not willing to support such projects. We looked at the concentration, whether we do it with filtration, with centrifugation as mentioned, or with magnetic beads. And we think that so far a combination of the methods gives you the right matrix with regards to speed, time, and cost. Is you have to combine something between the two, and ease of use for others. We looked at the other methods like the ddPCR, the digital droplet PCRs, and I think the cost is going to be prohibitive with regards to the outcomes expected. So, as this is mostly about multi-drug resistance, what we also were able to identify, which we thought was, we're just about to submit this work, is we've been able to identify some very interesting multi-drug resistant bugs, including Candida auris, from just wastewater samples, not from hospital-related samples, from community-related wastewater samples. And not only that, but recently there has been a publication in Singapore with a new clade for Candida auris, and we've actually been able to identify that in wastewater samples here. So we've been able to isolate, culture, do the multi-drug, the MIC, the sensitivity testing for the Candida auris, in addition to the whole genome sequencing to identify that it's actually one of the we have also identified the newest emerging clades. So overall, what we have experienced here, so we're a relatively new player in the field in comparison to many who have been doing this for hundreds of years. We actually are beginning to firmly believe that it probably is one of the most important tools for the way forward for monitoring whether outbreaks or trends, including multi-drug resistance, as it does provide a composite sample from a very large population that is probably the most cost-effective way of looking at things. We don't know yet whether the global standards will be. There are directions towards standardizing these methods now, and a lot of people beginning to identify these lacks and difficulties that we have to address very soon. The Europeans have already established a global pan-European wastewater surveillance program and project, which will be utilizing standardized methods, which I think, just like the UCAS before, a lot of us will end up contributing to and adhering to. I'll probably stop here, and we'll take the questions later on with the panel. Thank you very much.